This is an overview of an article entitled Rho Guanine Nucleotide Exchange Factor, ARHGEF17, is a risk gene for intracranial aneurysms, published in the July 2018 issue of Circulation, Genomic and Precision Medicine. Intracranial aneurysms, or IAs, are weakened areas in cerebral blood vessels that lead to abnormal dilation or ballooning. The danger posed by IAs is rupture, leading to life-threatening subarachnoid hemorrhage. The formation and rupture of IAs are likely related to a variety of risk factors, including genetic factors. These genetic factors remain to be identified. Attempts to use genome-wide association studies, exome sequencing studies, and family-based studies to identify IA genes have been challenging, in part due to the rarity of IA cases and incomplete penetrance. Here's the study design and the rationale behind it. For a late-onset complex disease, a suite of common variants will have modest effects on IAs and will be difficult to detect. Meanwhile, some rare or low-frequency variants will have larger effects on IAs and contribute individually or in combination to the risk of IAs. Those rare or low-frequency alleles with larger effect sizes can either contribute to the familial form of IA or act to increase the risk of sporadic IAs in the general population. To increase the yield of genetic studies of IA, one can aggregate the familial IAs and sporadic IAs to perform variant association tests to find candidate variants. One can also integrate sequencing data of unsolved IA cases from previous studies to replicate the candidate variants, as well as perform gene-level variant enrichment tests to prioritize candidate genes. In the paper under discussion, the authors genome sequenced 20 Chinese IA cases. They performed an initial variant filtering analysis and identified 30 candidate variants. This was followed by a replication of those 30 candidate variants using 3 independent cohorts, including samples from Japanese, European American, and French Canadian cohorts called Replication 1, Replication 2, and Replication 3, respectively. Finally, a gene-level variant burden test was performed on the 6 genes with variants rediscovered in the replication samples. On the left are the 30 candidate variants and the results of their replication analyses based on the p-values. Only 6 variants in 6 genes were observed in any of the replication cohorts. On the right are the counts of unique rare and deleterious variants found in cases, controls, or both for the 6 candidate genes. Only ARHGEF17 showed an excess of variants in the cases. A total of 5 ARHGEF17 missense variants were observed in IA cases. On the left is a schematic showing that each of the variants lies in a conserved domain of the protein. On the right are the pedigrees of the affected patients with ARHGEF17 variants, whether in the discovery or replication cohorts. There's evidence of segregation of the variants with disease in the families. The authors used zebrafish as a model to assess ARHGEF17 as a candidate gene for IA. In the left panels, in situ hybridization demonstrated ARHGEF17 gene expression in various parts of the zebrafish brain. On the right, the images established the expression of ARHGEF17 protein in zebrafish brain vessels. The red is the protein, and green marks the vessels. The authors used 2 different morpholinos to knock down expression of ARHGEF17 in zebrafish. In the top row of panels shown here, the middle and right panels show that each morpholino resulted in extravasation of blood, that is, intracranial hemorrhage, in both the forebrain and the hindbrain, as indicated by the arrowhead and the arrow, respectively. In the bottom row of panels, the arrows in the middle and right panels show that red blood cells, seen in red, have collected at sites of endothelial cells, seen in green. These images allow mapping of the bleeds to specific intracranial blood vessels. Here, in the images to the left, bleeds have been mapped to various sites in the forebrain, midbrain, and hindbrain. The graph to the right shows the results of rescue experiments. In the left part of the graph, the blue bar indicates the percentage of control zebrafish with intracranial hemorrhage. The orange bar indicates ARHGEF17 knockdown zebrafish. The gray bar indicates ARHGEF17 knockdown zebrafish in which the wild-type human ARHGEF17 gene was expressed. And the yellow bar indicates ARHGEF17 knockdown zebrafish in which human ARHGEF17 with one of the 5 missense variants found in IA patients was expressed. These results indicate that the wild-type human gene rescued the hemorrhagic phenotype, whereas the variant gene did not. The right part of the graph shows similar results with a different morpholino. In conclusion, the authors identified ARHGEF17 as a candidate IA gene with pooling of familial and sporadic IA cases for case control analysis, followed by attempted replication in multi-ethnic familial IA samples. They ultimately found 5 different ARHGEF17 missense variants in IA families. They also found that ARHGEF17 knockdown in zebrafish caused intracranial hemorrhage, which could be rescued by the human version of the gene. Together, these results provide strong evidence that ARHGEF17 is a risk gene for intracranial aneurysm.